You're listening to The Interactome, a podcast by a group of young researchers who want to connect you to the world of science by sharing their stories and perspectives. Just in case their bosses are listening, they want to remind you that the opinions expressed here are their own. They also want to remind you not to take anything they say as medical or professional advice, as they are not doctors. Not yet, anyway. Stay tuned about that. And, without further ado, welcome to the Interactome.
Hello everyone, and welcome back to the Interactome. I'm Sam. I'm going to be kind of running this particular episode.
Woo! Go Sam!
Woo! Woo!
I guess I can explain who I am and why I'm doing that, but maybe we should first have the other hosts introduce yourselves.
Hi everyone, I'm Maia. Yeah. It's been a long day for me.
Maia has an experiment running, right?
I do. Well, I just finished it, and then I was on the microscope, and I was like, boop, 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 I gotta finish, boop, 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 and then now I'm here. So I finished on time. I'm happy to be here, but my brain is catching up. So yes, but I'm excited to be here, to be on this episode. Yes.
And I'm Natalie. It's okay, Maia. We love you. It's all right. Do you want to do that again?
No.
Keep that in.
It's probably fine.
Let's keep this in too.
Yeah, seriously. I'm Natalie. I don't work in science. I am a climate tech and communications professional.
Yay.
Woo.
Yeah, so, and I'm, so, like I said, I'm Sam, and I do... try to think of the best way to explain this. I do kind of some stuff in pharma, with biotech stuff. I until recently though was a graduate student studying biochemistry and biophysics, and the important part that makes it relevant to this particular episode is that I have spent a very long time thinking about proteins. So this is the third and final part of our Central Dogma series. We'll get into why proteins are there at some point later. But I think the first thing we should discuss is what are proteins. So I was working at a biotech job, and one of my colleagues, who's a very smart person, he's currently in med school, we were working with proteins, but he was also pretty funny. He's also a pretty funny guy, and so at one point he just walks into the lab, goes, "The only protein I care about is whey protein," and just walks out. And so for most people, you know about protein if you're trying to bulk up muscle mass. Although you can't see me, I'm not particularly jacked. I have not consumed that much whey protein, but it's still the same stuff. But what you think about with proteins, like dietary protein, tends to be stuff that your body is breaking down to build proteins of its own. So your muscles are made out of protein, as are a lot of other parts of your body that you probably wouldn't think of being made of the same thing as muscle. Hair, the parts of your eyes that you see, the taste receptors on your taste buds, and lots and lots and lots of other things.
I thought that this episode was us learning how to get ripped and gains. All the gains.
Protein.
Yeah, I don't think you want to learn that one from me.
Protein.
I did the wrong kind of preparation for this episode, everyone. No, I'm just kidding.
Maia's doing deadlifts next to the microscope.
In my notes, I have a gym regimen. I have diets.
Hey, I haven't been in the same room as Maia for a long time now. You might be way more ripped than you look on camera right now.
Grad school changes you. You need to find a hobby that exercises your brain and your body in several ways.
A lot of people do work out quite a bit.
That's true.
I definitely did work out more in grad school, but I wouldn't call myself ripped. I don't think you want advice from me.
No, but I think that's kind of cool how you mentioned that the proteins that we eat. So that would kind of be meats and things like tofu, I'm assuming. You eat those proteins to help build more proteins, and I think that's pretty metal.
Yeah, protein's also in a lot of.
Maia
other
has
stuff,
the
too.
giggles today.
Yeah.
Maya has a case of the giggles.
Hee hee.
And I'm living for it.
Thank you.
So, yeah. So the thing about proteins, like I was saying, they have a lot of different functions. And why proteins are able to do all of this is because of the fact that proteins are made out of more building blocks than the rest of the stuff we've been talking about before. So when you talk about DNA and RNA, we're talking about 4 different building blocks. We're talking about nucleotides. Proteins are made out of amino acids, and there are 20 odd amino acids. I think it's 21. But
22. There's one for almost
20 natural.
every letter of the alphabet. So like
20 natural, and God knows how many more.
synthetic.
It's not necessarily synthetic, but your body modifies them quite a bit. So, but we talk about 20 amino acids. So those are 5 times as many different options for chemistry than you have with the 4 nucleotides that you have in RNA and DNA. They also have very diverse shapes. So you can have ones that are, so these amino acids, they're ones that are just little chains. They're pretty much have nothing going on. There's other ones that have giant ring structures and things hanging off of them and can serve as places to tack on even larger structures biologically. And what this means is that you can have a bunch of different functions that come from that structure. Those structures can be really, really complicated. So proteins like DNA or RNA are synthesized as chains to begin with. So just kind of stick one thing to another, like stacking building blocks or stringing beads on a chain, I guess. Like you really just have an ordered set of things. But very quickly, something else happens, and that's what we've referred to as protein folding. And so protein folding makes these complex 3-dimensional structures, and structure defines function. So you can imagine that the same proteins that your body uses to make muscles are going to look very, very different from the ones that, say, allow you to see. And structure defines function. You wouldn't try to write a letter with a doorknob or a dump truck. And that's kind of the same rules apply biologically. Some proteins are really, really big and some are really, really small. Some are relatively inert. They don't really do very much. They just form structures, while others contain very complicated assemblies that can do things like spin like rotors or move like levers, or some of them even just kind of walk. They're actually quite funny looking. So the reason that all these structures can happen from these 20 amino acids is this thing called protein folding. And that happens of its own accord. We think, for the most part.
Sometimes,
most of the time. And there's actually was this grand debate the second that was mentioned, because like most things in biochemistry or biology in general, the second somebody opens their mouth and suggests something major, someone else wants to fight them. So the guy...
Classic.
I've been in the room for some of these. Not quite that major, but people have very strong opinions and will duke them out. And that's just kind of how it goes. The guy who proposed that protein structure comes purely from its sequence, so purely those order of amino acids on that chain, a guy named Christian Anfinsen, who proposed that protein structure is dictated by sequence alone. And so this means that that sequence of amino acids linked together, which we call a polypeptide, which is funny. You'd think you'd call it a polyamino acid or something, but we call it a polypeptide because of the bonds between them. They're peptide bonds. Anyway, he proposed that a polypeptide should be able to fold itself into the structure it's supposed to occupy with no help. And that is great. And a lot of times you can see that in a test tube. So you can take a protein that's already folded, you get it from nature, you put it in some chemicals that make it unfold, and you take it out of that environment, and a lot of times it'll just pop right back.
I think that's really cool.
Yeah.
It's just like, it's reversible. Yeah.
It's also such an important evolutionary tool that proteins have, right? Think of how often, if our proteins were, say, really sensitive to temperature, right? And we couldn't.
But they are.
Oh, am I giving?
But they are.
Am I getting?
We'll get there.
Sam literally just got the biggest grin as soon as I said that. So I'm gonna take a step back. It sounds like he has something.
No, I think you made a point, because the last episode
yeah.
we talked a lot about primordial soup and how like conditions were like crazy, but it could be related to that.
Some proteins, which we'll get into a little bit later because they're wild, actually won't unfold even if you essentially throw them in an oven. or boil them, and other proteins will fall apart of their own accord just because they're sitting there. And if you heat that up, they'll do that more.
Oh, I have a fun fact about a really stable protein.
Ooh.
Oh, so
Which...
someone in my lab studied the protein SOD1. And while she was doing like her like review of the literature, to like write the introduction of like the thesis and the paper and everything, she and my PI came across this paper that said that they were able to isolate a SOD1 protein from a mummy that like people found that was just like, being dead for like lots of years. And so they were able to isolate like the SOD1 protein from this mummy and then kind of like, pamper it a little bit, like put it in some buffer, and it's still catalytically active. So these guys can be pretty stable.
Yeah, and what's wild is like, I work with some things that are very stable too. So typically when you talk about like biotherapeutics, drugs that have proteins in them, you want them to be very stable. The wild thing, though, is when I say, they are sensitive to heat, is so what I did in grad school was I studied what are called chaperone proteins. And chaperone proteins are proteins that help other proteins fold. And so Amundsen goes and says, hey, these proteins can fold of their own accord, and then your body has all these really essential proteins that their entire job is to help other proteins fold. And you kind of go, what's up with that? And part of it is it's a protein in a test tube is not a protein in a cell. And yeah, it's a bit like if you were trying to like solve a math problem by yourself in a nice quiet room, or if you're trying to solve it in a kindergarten next to a construction site while a police car goes by. It's loud, it's chaotic. It's not loud, but it's chaotic,
it also goes to say too that like the conditions in a lab are very rarely, if ever, the actual conditions that a biological process or a chemical process, etc., will have to take place, just because there are so many unknown factors. I just wanted to add that to
this is the source of a lot of heated debates in the science community. Like, what's the best like way to study something? What's like a good model system? Does it actually recapitulate the biological process and the environment that it's taking in? You're absolutely right, Natalie.
And the way you can sometimes test that in the lab is just making the environment more viscous. You just put in goop. And
really?
Yeah, you just put in glycerol, which
goop
is like found in a lot of like cosmetics and things. Anything that needs to be kind of goopy.
Why goop it?
Because it crowds the environment, makes it like a cell. So it's
RNA
like...
soup, protein goop. I'm sensing a theme here.
We are simply goop.
Yeah. And one of the hypotheses for why chaperones are important is just so that the proteins themselves don't become goop. So
I do like how they're called chaperone proteins. Sorry, as a side, like maybe to make it clear, it's like, how like chaperones are, like to go back to your kindergarten analogy, like, like you have a parent chaperone that like makes sure like all the kids are like behaving, that kind of thing. I guess like analogous to what happens in cells, the chaperones are like the parents like making sure the little kindergarten proteins are like folding and quote unquote behaving. Okay.
Yeah, that's great. That's a great analogy. I love that. The one kind of macabre problem with that analogy is a lot of chaperones also send proteins to the garbage disposal.
Sometimes there's tough love out there,
So,
protein parenting.
I don't question parenting styles.
Well, it's like proteins, you just grind them apart and make new ones again. I mean, your body started with grind up protein anyway, or broken up amino acids anyway.
So if you have a misbehaving child or protein, you banish garbage. Let that be a lesson to you, children.
Well, I wasn't expecting this to go into me being like, well, it's actually shredding. I'm like, yeah, we're just going to just leave it there. Not really shredding, but they'll take things apart. There's proteases. It's funny. Proteins are helped and disposed of by other proteins is great. It's proteins all the way down. So now we've talked about what proteins are. I guess it's like, where does this thing that needs to fold come from? And yeah, so we can back up to that. And frankly, that's less my area of expertise. I think y'all probably studied more molecular biology than I did. I tend to start with a protein, go, what does it do? And
No, I feel like you touched on all the right parts. Like, yeah. So, like the central dogma that we've been referring to is kind of this, like, how do I describe it? Like a quote unquote like canonical process for how proteins are made in the cell and how information is conveyed through like different molecules to like produce the proteins, which we know, as Sam described, have a lot of different structures and therefore functions and carry out a lot of like the important things that the cell needs to do, whether that be like, you know, like muscle cells doing their thing or proteins in like your cells in your eyeballs that can let you see. We talked about this in a few other episodes, but to kind of like breeze over the first couple of parts of the central dogma, all of our genetic information is stored in DNA. And this information is transcribed into RNA, which is kind of acting as our messenger molecule. So it's kind of like the go-between of like the DNA and the end result, which is the proteins. So the RNA molecules have kind of like a copy of the genetic information that's stored in the DNA, and this genetic information is instructions for how to make a protein. So the instructions for the proteins are basically these 3 nucleotide sequences called codons. So you can find those on the RNA in like discrete, like 3 nucleotide or letter chunks. And they're called codons, which I think is kind of cute because they're like part of the genetic code. So each codon corresponds to one amino acid. So like one of the little bits of the protein that Sam mentioned earlier, that can be kind of like put together. They have like different shapes of their own, and this allows like a lot of different combinations for chemistry in the protein backbones and for the proteins to like fold and adopt their shapes. So the RNAs have the information for the codons, which correspond to the amino acids that make the proteins. And basically, the RNAs are specifically, in this case, messenger RNAs. We talk a lot about several different kinds of RNAs that exist in the cell in our very fun RNA episode, the one right before this one. But basically, the messenger RNAs are kind of fed into these little protein-making machines called ribosomes. And they're kind of fun. Like, structurally, they look like little, like, mushrooms, almost like little balls that like sit on either end of like the mRNA and just kind of like chomp on them, if you can like kind of picture that. And basically they catalyze a reaction in which the mRNA codon is matched with another kind of tRNA, with another kind of RNA molecule called a tRNA or transfer RNA. And this tRNA has the specific amino acid that corresponds to the codon on the mRNA attached to it. So the ribosome kind of like facilitates this matching up of the mRNAs and the tRNAs with the amino acids attached to them. And then as like the ribosome is kind of like reading this like long string of mRNA that has all sorts of different codons, different tRNAs with different amino acids attached to them are also coming in and like docking on the ribosome. And this is where also all of that chemistry that Sam mentioned happens as like these different amino acids are getting kind of like linked and catalyzed together to form kind of like the first kind of like preliminary, if that's a word, primary is a better word, structure of the protein. So that part is called translation. So the mRNA is being translated into a protein. So the ribosome is just like zoom, 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 like moving along the mRNA, translating it into a polypeptide chain, which is the primary form of a protein. And then a bunch of spicy things happens after that, where like folding and like chaperones can come in, so that the proteins can adopt their proper structure. Molecular Bio 101.
So the thing that's always amazed me about ribosomes, though, is they're not proteins.
They're made
Right?
up of a bunch of little proteins and RNA. They're both.
Okay.
Yes.
Because I just know about like the ribosomal RNA. They
Yes.
refer to it.
Oh yeah, that one is really important too. Yeah.
And so like that kind of goes back to your soup,
Yes.
your primordial soup,
Mm-hmm.
because it said it was proteins all the way down. That was not entirely true. The thing that makes proteins is not kind of entirely right.
Yeah, it's like both RNA and protein, and it has interesting evolutionary kind of like hypotheses surrounding like why is there ribosomal RNA in there mixed together with the proteins, because people think that the ribosomal RNA has catalytic properties, which catalyzes the peptide bond, the amino acid like chain linking together.
Do they think it came first?
That's the debate.
All right.
Mm-hmm.
So that's a very thorough recap of how translation happens. That was way better than I could have done. Considering the fact that my first task in undergrad was describing the transcription and translation, and I wrote the wrong one for each.
They're
People
very
certainly-
similar words. Honest mistake.
So if you're confused, don't worry. I have been too. It was great because I was in it was like I'd taken an advanced science class in high school, so they were like, oh yeah, you can skip to this one. And so like my first act in the advanced biology class in undergrad was to completely confuse those two words. My professor made me rewrite the whole thing. It was great. Just kind of old school, but
I just know because I just know some stuff about translation because my very first lab was a translation lab, so I had to know it or else face
Yeah.
the consequences.
yeah. So yeah, so we kind of going back to the folding thing. So you're saying like, oh yeah, we have this primary structure. And another thing a professor had me do was not confuse that with primary sequence. So I will never ever call it primary sequence because that is a pet peeve of someone who I took a couple classes with. But so that folding question, which is a thing that's going to get harped on over and over again because that's all protein biochemists care about at the end of the day. I mean, we care about other things, but the folding is what protein biochemistry is focused on since its inception, is what that fold looks like and how does that influence things. Going back to Anfinson's whole, the structure, the sequence is all that matters. So Google recently, in the past 3-4 years, made an AI called AlphaFold. And unlike the image generation AIs that you ask them to hey, I want a picture of a duck and it makes a duck with 3 heads or whatever because it doesn't quite have it. AlphaFold, you ask it, hey, here's a protein structure, protein sequence, give me the structure, and it can actually do it. And this was not something that was a given. It was something that when I started undergrad, which was not that long ago, it was kind of a dream, maybe we could possibly do that. Maybe Anfinson's right. And Google went ahead and demonstrated that to the point where now a lot of people will throw things into AlphaFold to start trying to get kind of a picture of how things happen.
And AlphaFold is very cool, and might I say, a lot of people like to joke that it threatens the livelihood of every structural biologist because a lot of people spend time trying to figure out structures of proteins of interest, the things that they're studying, but then now that we have AlphaFold and we can just boop boop put in a protein sequence and it makes in some cases a pretty high confidence guess of what the structure looks like. It's do I even need to try to solve the structure of this protein on my own when AlphaFold can do it?
Yeah.
But it's a certainly very useful tool.
Oh yeah, and those ways of getting structure take time.
Mm-hmm.
I've actually never, I don't know if you've had to do it, Maya. But...
I'm currently doing it.
Ooh, fun! So I've never actually had to do it. So maybe you could talk about, there's a few different methods for getting structures. So, Maya, do you want to talk about what you're trying to do?
Yeah. So, Yeah, so basically, in my thesis project right now, I'm a grad student. I am dabbling in some protein biochemistry myself, having a lot of fun, learning a lot of things that we're mentioning in this episode. But basically, I'm trying to get a structure of a protein complex that is unusual. So no one has ever kind of like defined it before. And we're trying to figure out a structure so that we can better kind of, like Sam was saying all along, figure out the function of this protein complex and better understand what it might be doing. So to do this, we're using a few structural methods. So the protein structure that I'm studying is called the proteasome. So it's like actually one of those proteins that we were talking about earlier that like does the degrading of like the garbage bad kid proteins that we were speaking about earlier. But yeah, my thesis is on that garbage can machine, the proteasome, that does all of the degrading. And specifically, we're interested in interactors of the proteasome, so other proteins that can combine to like the proteasome kind of like trash can and change how it might behave. So if you imagine like the proteasome is like it's kind of like exactly shaped like a can, so like a garbage can, I suppose, but it's shaped like that, and it can have proteins that sit on either side of the can that kind of regulate what proteins go inside the trash can and get degraded. So I'm interested in studying one of those interactors that's understudied and like people don't really know what's going on. As an interesting side note, this interactor is heavily upregulated in meiosis, which is the biological process that my lab studies. So it has some kind of like physiological / developmental implication. We just need to like work on it a little bit more. But anyways, we're interested in kind of solving those little proteasome different interactor structures. But this is kind of hard because the proteasome is like really big. So it's a very large protein complex. It's made up of, I think, around there's at least like no, I should like cut this out because I should know this, but
it's fine.
I was like, oh no, professor, please listen to me. But
well, Maia, things are dynamic in cells.
Things are very dynamic.
So whatever number you say, it's probably going to be wrong.
and you know, people could always prove me wrong later. Discover more, discover less.
Oh, I'm gonna say there is no one number, is what I was trying to say.
Okay, perfect. But it's made up of several proteins, and it's really big. So you could imagine that if you have like one really cute little tiny protein, it's easy to kind of like know what it looks like. But when you have a protein, or depending on like what it is, we can get more into that later. But if you have like a whole complex that's like really complicated, that has a bunch of proteins in it, it's very complex. Exactly. It can be a little hard to do that. So we're using a structural method called cryo-EM, which is all the rage recently in structural biology. Back in the old days, and people still do this for some proteins, but they used to crystallize proteins, so they would purify them from like cells or plants or like whatever source you get them from, and they would like kind of like work away day and night to find these like optimal crystallizing conditions that like the protein that you're studying would like make these like fun little crystals, and then you would take that to like the X-ray crystallography room, and then you would like bombard it with like X-rays. And the cool thing about that, because like you might be like, okay, we have like a crystal, and like what are the X-rays doing? Like how do you get the structure of like your protein from that? There's like a lot of crazy like math and biophysics that goes on, and like Sam, you probably know more about this than I do, but basically like from what I know, like the X-rays kind of like hit this crystal, and then like they diffract in like crazy patterns, and then like a computer kind of like gets all of the information on how the X-rays are diffracting after they're like hitting the crystal that you like worked so hard to get. And then some like math happens, and then
And
you
then
can, like,
math happens.
and then math
Then math
happens,
happens.
and then you can like take that diffraction pattern and like match it up
Yeah.
to like a structure.
Yeah, so I think maybe we should back up a second and go, why can't you just look at them under a microscope? And
True.
and so first off, you can kind of. So cryo-EM, which Maya mentioned, is short for cryo-electron microscopy. But you'll notice that there's that electron there, and that's a huge difference. You're not going to stick an electron microscope in like a kid's science kit, at least not currently, although I do know someone who tried to... He had a scheme to build one out of an old TV. It's a whole thing. But anyway.
But those microscopes are expensive.
They are. And
Why are
yes.
they?
Well, I actually don't know whether it's super expensive, but I mean, in general, we
Or how are they
could back
different?
up. So a light microscope works with light, the visible light around us, right? And this is a wild thing to get into, but essentially you see things because light is either bouncing off of them or being refracted or reflected, or any number of things can happen to a wave of light, and provided it makes a change that your eyes can perceive, then you know a thing is there. The thing about that is it has to be big enough for light to interact with it. So there's a rule with microscopy, which I think is like you can only see things that are larger than half of a wavelength of light. So light is weird. We don't need to go too deep into it. I'm not an expert on it. We could get a physicist in here and really explain it, but light travels in waves, and those waves have a wavelength. So just like if you have a wave on the ocean, there's a peak and a trough, and you can kind of say, oh, there's this much of a distance between them. And the wavelength of light is in the hundreds of nanometers range, the light that we use to see as humans. And proteins are usually in the tens of nanometers of size at most. And what this means,
those tiny guys.
they're they're so tiny, which means that anything smaller than like a bacterium is actually too small to see with light. It won't interact with light the way we like things to in order for us to be able to see them. And so what instead happens is we have to hit them with something that has a shorter wavelength, and which corresponds to higher energy. So you're essentially building a super powerful ray gun. It shoots electrons. It's an electron cannon. And you shoot it at a thing, and you have a very sensitive detector that says, hey, here's an electron, here's not. The thing is, you can't necessarily measure with the resolution of needing, like, seeing where each electron goes. So the best trick to get that to work is make a crystal out of it. And what a crystal is it's just a repeating pattern. So instead of taking a picture of one protein, you're taking a picture of thousands or millions or however many proteins all stuck together in a repeating pattern, like an MC Escher painting or a tiled floor or whatever. It's the same pattern repeating over and over and over again, getting hit by by a gun of electrons, and then they're scattering the same pattern. The thing is, proteins on their own also don't form crystals, typically. Most proteins. There are exceptions to this, but you need to trick the proteins into forming a crystal that is regular enough that you can shoot an electron at it and it will scatter consistently. And there's like different amounts of luck that come into this, so you get different resolutions, which means you get a blurrier or better picture of a protein. That takes months because it's a lot of like, there's people who want to crystallize proteins usually will just put a bunch of little tiny containers and put a bunch of different stuff in them and shove them in a room and hope you get crystals.
Yeah, it's like to maybe put it in like a perspective, like if you've ever kind of like tried to make rock candy as a kid, it's like kind of like that, but like with like 20 times 100 times harder.
No guarantees, and it's just a lot of throwing stuff at the wall.
Mm-hmm.
I worked next door to a former crystallography lab. I've worked near them, and they have the randomest chemicals because it's just throw things at it and hope it works. And this means it'll take like months and months to get a single crystal, and then that crystal's super precious, and you don't want anything to happen to it. But there's another trick you can do to get the same structure over and over again, or at least keep proteins from wiggling, because they do this. And so the crystal keeps things from wiggling for the most part. You can also just make them very cold. If you make something cold enough, it stops moving. If you make me cold enough, I'll stop moving too. So so there's this trick of just making it really, really cold and then shooting it with a beam of electrons again. But this time we're doing what's called electron microscopy, which is usually better for bigger stuff like circuit boards or the surface of an insect or a bacterial cell. A cell, for example, is big enough to take pictures. Cells have millions of proteins in them, way bigger. The trick with cryo-electron microscopy, or cryo-EM, is you just take a bunch of pictures and then you have a computer average them, and that makes it less blurry. So that's also neato if you're doing what Maia's doing, where you have these big complexes, because big complexes don't love to make crystals and repeat themselves competently, or even be the same over and over again. And so you can have a computer go, this one's like that, this one's a little different, and at least that's my understanding.
That's perfect. Yeah, I have to graduate one day. So
yeah.
I can't be trying every combination of chemicals and conditions. And the lab that I'm actually collaborating with a lab on this project to try to get the structure of the big proteasome complex that I mentioned. And they've gotten it figured out nicely, which is good for me. Not like the crystallization conditions, but like kind of like the workup to cryo-EM and like what to look for once you perform the cryo-EM.
Yeah, and there's other weirder ways of getting structure too. So you could also shoot things with X-rays and not freeze them and not make them into crystals. So I actually was kind of in a lab that did that for a little bit. But you also do that to determine how wiggly things are. You can also do something called NMR, which is nuclear magnetic resonance, which is not involving radiation. It's just involving giant magnets, and you make things spin a certain way and then you make them spin another way. And I somehow, by determining the change in the spinniness and things that give off radio waves, you can get structures. It's bonkers. All these things get really, really weird very quickly. The funny thing about NMR is it's also what we call an MRI. When you go to get an MRI scan of your body, if you have like an injury or an illness or something, it's nuclear magnetic resonance imaging. They just knock the nuclear off so people wouldn't get scared of it. Nuclear in this case refers to the nucleus of an atom, not the nucleus of a cell, and not nuclear radiation, like that sort of thing.
Good distinction to make. Yeah.
There's a lot of different things that have the word nuclear in them. Your nuclear family is also not radioactive.
Let's hope not.
Hopefully.
You never know.
Anyway.
No, lots of cool ways to determine structure, and it's great that we have AlphaFold now that can help us with a lot of that.
I think the cool thing about proteins too is that like what we've learned from this episode is people will dedicate their entire careers to studying one protein because they are so complex. Sam's covering his face right now. He's getting flashbacks.
Oh boy, what I decided not to do.
Yeah, exactly what you decided not to do.
Mm-hmm.
But it's very cool. They're very cool. I very much respect people doing that. I depend on people doing that.
Yeah, and there are whole fields about like individual proteins. I think there's like whole conferences about like specific proteins, which is like kind of funny. But yeah.
I just want to touch on something too that Maia brought up at I think towards the middle of the episode. You mentioned upregulation. I think that's one really valuable part of just, I don't know if you can call it like the protein life cycle or protein like existence, but when proteins are existing in an environment, in a cell, certain conditions call for more of that protein or less of that protein, and the cell has the ability with, signaling and all that stuff to be able to, we don't need that much of that protein anymore. We do need more of that protein, and kind of adjust on the fly, which adds kind of another layer to that complexity.
Yeah, you're totally right. And like to kind of like tie it all together, like the way that like for example, like if a cell is like, oh my God, I need like more of this protein at this time, the whole central dogma or maybe parts of it can kick in and contribute to like the production of more of that protein. And then like maybe if you need even less, like you were saying, the protein can get like degraded and shredded up, or there can also be layers of regulation that happen to parts of the central dogma that make them like not as fast or efficient. Therefore, kind of like putting a damper on like the protein production.
Yeah,
as
it's like
well.
a yellow light almost.
Yeah, yellow light is like whoa,
Yeah.
whoa. So that's all like very exciting, and it's really cool how like the cell has all of these kind of like ways to do that and adapt to like different situations, like you were saying.
And the fun part too is these regulations, the red light, green light, yellow light, is also proteins, DNA, and RNA. It's all made out of that stuff for the most part. And there are sugars and fats that come into this too, primarily more sugars that are like, those are other macromolecules and they do other things. They're not just things that go into a good cookie. They're molecules that have their own functions and things like that in the court too. But like, you know, you have these, like, upregulation could be, hey, there's this protein that noticed a thing, and that is going to bonk into something, another protein that's going to change something about DNA, and then that's going to make different RNA, or change what RNA is sticking around, and all these other things, and it's very, very complicated, but it's kind of a lot of the players are the things that are also being regulated, which can lead to these terrifying diagrams.
So if you're listening to this podcast right now, you should know that your proteins and your DNA and your RNA are all doing great jobs. So much can go wrong. Hopefully, knock on wood, and hopefully they will continue to do great jobs. But it's kind of like, honestly, like when I think about it, it's like kind of a miracle that like any of us are like alive and like functioning. Because I'm like, there's so many things that could go wrong, and like the more you study biology, it's like, oh, so many things can happen.
And
so,
you know, listeners, don't get, you know, anxious about that, right? Our body has a bunch of checkpoints to make sure that that stuff doesn't happen. And then even Sam's trying to whisper, what is it? I'm nervous.
If you do get anxious, that's also proteins.
No, true. There's no escaping.
Sorry. That was
All
very
good.
dumb.
there's no escape.
There's no escape.
No, but yeah, proteins can really do so much of everything, and they have so many cool shapes. One thing that I wanted to mention is that, and maybe this is like a tool that you've seen before, Sam, probably definitely, but there's a website called the Protein Database, or PDB for short, where people deposit like the structures they've solved after, you know, long nights in the lab or at the microscope, that kind of thing. But it's a repository for all sorts of protein structures that labs have worked on. And it's like kind of cool to just kind of like click through it. So something very cute that they do is that they have a molecule of the month. So I think like a favorite structure gets voted into being like the molecule of the month. And then I think
That's
like
adorable.
Towards the end of the year, it's so cute. And towards the end of the year, I think they like have put together calendars of all of the molecules of the month. So you can like look at this on the protein database, but there's a lot of proteins that have really neat structures like donuts. So they have like holes in the middle, maybe to like let things through or like attach to something. A lot of proteins have like really cool symmetries or symmetrical properties. So like if you've ever done that thing like when you're a kid where you like paint on one half of a paper and then you like fold it over and then it makes like a cool kind
Like
of
a Rorschach
art
kind of
yeah,
looks.
Yeah. So there's like a bunch of proteins that have like interesting symmetries going on. And then you can even look at like what viruses look like and all like their different proteins and things like that.
Is that 3D printed, Sam?
Ooh.
Yeah, it's a 3D. It is a structure from the PDB that I convinced the people to work to print for me.
Nice. So Sam has his very
presentation.
own molecule, special molecule. Why don't you describe it for the fans?
I mean, I don't know how much work once we go into everything on this podcast, but it's a structure of a protein complex that I work with. I'll leave it at that. And
it kind of looks like something you throw in your laundry to get rid of like lint.
It does.
Yes,
It does.
A lot of protein things look like that too. But it's really nice. The protein database now has a little thing you could like click a little button and it'll download a 3D printer file for you.
Oh, that's really cool.
Yeah.
Oh, that's so fun.
If you want one on your desk, if you happen to know someone with a printer, we happen to work somewhere that happens to have them. It's very cool. Yeah. I had like snuck away to grab that. It's not actually. It was a visual gag on an audio medium.
It's really cute. It's just like a.
We're technically recording Zoom right now too.
we could just like
see that.
redacted.
Redacted.
No, but you can imagine Sam is holding a very adorable palm-sized protein that looks like it could be a dryer ball, maybe. But no, that's life.
one of my, and as we're coming up on, the end of the episode, this is one of, when I learned about what we're going to talk about next, I'm like still obsessed with them to this day. Protein folding can go very, very wrong.
Yeah.
And if it goes wrong, there are different agents that can cause it to go wrong. I'll open the floor. But we're about to talk about one of my favorite things in biology ever.
I'm glad. It's one of my favorite things, too. It's the protein fold that kills you.
Yay!
So we could back up and talk about, like Maia was talking about, the proteasome. Why do you want to get rid of old proteins? There's a lot of different reasons for that. Sometimes they just don't work well anymore. But a buildup of junk in cells is not good. I was actually doing some random Googling recently, and that's like, why asbestos is not good for you. It's just buildup of junk. It just happens to be kind of like glass, but it can also happen with proteins. And so proteins like to form these nice symmetrical structures sometimes. And sometimes proteins go wrong and they still form a nice symmetrical structure. And sometimes that structure is very, very, very stable. So one lab that I'm familiar with, like that lab I was talking about that did some funky stuff with x-rays, they'd shoot x-rays from like brain samples. They'd like shoot x-rays at like brain samples and things of people who may have in life had this sort of disease. And why this kills you is still a matter of debate, but we call this badly folded protein that forms this big complicated structure, we call that a prion. I think it's prion. One of them was a bird. Prion and prion.
There's when
Prion.
You Google prion or prion, both like the protein fold that kills you come up, and also like some species of bird in the southern hemisphere.
I've always pronounced it and heard it as prion.
Me too, but I
Yeah.
think both are acceptable.
Maybe.
Debate.
This is another one of those. We really need Lauren on here to really like assert something. But yeah, it's pretty much just the protein fold that kills you.
Because it's
thing,
like
though-
it's like really like goopy, well, for lack of a better word, like aggregating and like sticky, and just like messes up with like stuff that's happening in the cell, right?
Yeah, and why that matters is a little bit puzzling. Okay, you'd think that if you have a bunch of junk somewhere there's not supposed to be junk, it's not going to be good. This is a source of fierce debate. I have seen people get into heated arguments about non-profits that you probably think are wonderful. I have like opened my mouth and said I worked on one thing, and people like all the eyes in the room turned to me like, I swear I have no words in this place.
They're like, why are you offensive?
So I'm not going to open that can of worms any more than we already have. But there are a few diseases that we know are prion mediated, like it happens because of a prion. The one that is most well known is referred to as Creutzfeldt-Jacobs disease, which is the proper name for what people call mad cow, which is a infectious prion typically obtained from poorly handled meat. If there's like, usually I think brain material, it will eventually clog up your brain with garbage, and you don't.
Yeah.
Yeah.
I think the
It's
reason
not
why they
good.
called it like mad cow is because since like the prion of like the protein that's responsible in the disease is like you're saying, like aggregating and like clumping up like in the cow's brain, then it causes behavioral changes in the cow. So like people are like, oh, like my cow is like angry and like aggressive, and it's like not behaving normally, and it's because of that prion. And then and like you're saying, when like the meat is like improperly handled, like maybe some bit of the brain ends up in your burger meat, then there's a chance it could be passed to you after you ingest it, which is kind of crazy because like it survived kind of like being outside of like the cow's environment, and then it's like kind of like in the meat environment and like the processing environment, and now it's in like the person environment.
You can't cook it out. You can't even necessarily autoclave it out unless you're really, really thorough. So autoclaves are like the industrial sterilization equipment used in hospitals and labs. And there are very specific settings you need to use to kill prions.
So scary.
You need to heat it up very, very hot for a very long time. And it's terrifying. On the other hand, it's not the sort of thing you'll get from someone sneezing on you. It's also fairly well controlled because people know this will happen. There is currently a prion disease, I think, bouncing around in deer populations in the US currently, I want to say.
Didn't know that. Ooh.
I didn't either.
Yeah, I don't think it actually infects humans. I haven't heard anything about that. Something, I think chronic wasting disease or something it's called. I forget what it is, but...
Oh, do we know why it can't infect people?
I don't think we know that for sure.
Interesting.
I just haven't heard of human cases. I have not been following this very closely.
Yeah, because I never knew about like the like mad cow disease. Like if it was like rather that particular prion was just like very, very infectious, or maybe it was like similar to like something that you would find in people. I'm not sure if that's known, but there's like no cure, right? So.
Oh yeah, there is no cure.
Yeah, I remember being really... I think this, like, happened, like, when we were growing up, like, we were elementary school kids. Like, this was, like, all in the news. And I remember being very sad about, like, not being able to go eat burgers with my family.
Oh, yeah. I was afraid of burgers for a hot second as a child. But I don't think there have been many outbreaks. The other terrifying thing is it takes a long time for it to happen. But, again, people are very aware of this. As it was an issue in, like, the UK in the early 2000s, but even then I don't think it was super widespread. It's one of those things that's relatively hard to get, but particularly terrifying, and so it captures people's imaginations and anxieties. But there are more common, potentially prion-related diseases. So we think that Alzheimer's and Parkinson's may be prion-related. I say this, and if this ends up in certain circles that I've been in before, I'm going to have people yelling at me on whatever social network they find me on. I said may.
Hey, I feel like the wording. Yeah, I feel like the neuro aggregate people are very like strict about the wording. So I've only ever heard it be described as aggregates, but
they are repeating patterns of misfolded proteins. There are probably non-pathogenic versions too, which is weird.
Yeah, because that's what I thought. Like what helps distinguish prions from like other types of protein aggregates is that they can kind of like impose the misfolding on other proteins.
I don't think we know for sure where that line is, which is part of why like brain surgery involves a lot of sterilization stuff, as best I understand. And so like there's a lot of like care taken to try to prevent these sorts of things from being transmitted between people in a hospital environment too, just because we don't have a great grasp on exactly where that line is. And not to mention there are transmissible ones, and so if you're in an environment where you want to maintain sterility and safety and things like that, you just kind of assume that it's there. You act like it's there and treat it like you have to get rid of it.
Which is the smart move, I think. Always good to be careful.
Kind of like COVID, right? You act as though it's there.
If you're working in an environment where you're worried about infectious disease or potential risks, you always act like it's there and then take the steps to get rid of it, even though it's probably not. And that's, a great way to try to ensure safety, because if you act like it's not there and it is, well, then you're in trouble.
But definitely very interesting, and I think merits a lot of future study. It's like, what are they doing and how? Like, it's so like we don't know. It's so interesting.
So
yeah.
We need to know.
There are so many problems out there like that. If we want to wrap up, I mean, it might be fun to mention some of our favorite proteins.
I don't have any. None come to mind. I panic.
None?
Prions come to mind for me because they're so cool, but I don't. I was like, before this, I was frantically Googling cool proteins. But I'm just gonna say this is real, this is me, nothing comes to mind.
Is your favorite protein whey protein?
Honestly, yeah. Let's go with it. Shout out to the guy at the beginning of this story. That's what I'll go with.
All right.
I'm just getting that disappointment out of the way.
I really hope that your friend, the whey protein guy that went to med school, I really hope he becomes like kind of like a nutrition doctor or like muscle doctor or like that kind of thing. That would be iconic.
I feel like he's going for something way like, I forget what he's going for. I think it's like some like hardcore like surgical or something shit, you know, like he's a bright dude. He's also just got a great sense of humor. Yeah.
Love it. Yeah, I think I already talked about, I talked about the proteins that I study, so I'm kind of biased there. The one that I'm most interested, it looks like a little party hat. It's like a cone that sits on the end of the proteasome, like kind of like trash can surface. So I'm trying to figure out like what that little hat the proteasome wears is like what is its function, what is it doing, and how is it's affecting the rest of the proteasome. But I also want to mention another cool protein that I learned about was at a conference last year. And I can't remember the specifics about it. I think it was like a protein from a virus. But the reason why I liked it is that they solved a structure for it, and it looks like a scrunchie with like little beads on it. So it just like looked really cute. And then I really liked it because I had a scrunchie at home that looked exactly like it. So very adorable scrunchie. I love protein. That. Mm-hmm.
That's cool.
Yeah.
I think my favorite protein is, honestly, I think the first one I ever learned about in like freshman year of high school biology class.
Oh, you never forget the first.
You never forget the first. Yeah. And that's a protein called ATP synthase. So we have these energy molecules in our cells that we talked about a lot in the mitochondria episode. And I think this protein even came up there, but it just looks like a little rotor. It's cool because it looks like a rotor or a turbine, like you'd find in like a hydroelectric power plant or something. And it looks like that because that's pretty much what it is. And it's really wild that we have these structures in our cells that look like machines that people build. And so ATP synthase will always be like my favorite. Maybe I can see if I can print me one of those.
Yeah.
Maybe it'll spin around.
It goes burr. No. I feel like if you look up a YouTube video of ATP synthase, it's very hypnotic watching it.
It's a good time. It's a good time.
It's just like turning and turning and turning.
Just churning out ATP all the time.
Mm-hmm.
Your whole life. Just
whole life.
Jillions of those
in
your body. Just churning out that. And it's cool. I don't know. It's pretty.
Yay. But listeners, you too can find your favorite proteins. Feel free to check out the Protein Databases, or PDB's vast collection of fun structures. Or feel free to Google into more of the wonderful world of prions.
Yeah. There's probably some less nightmarish ones on the PDB. You know, it's like, hey, here's a little protein that's from like a mouse, and it, you know, so does cute. Mouse things. And you're like, okay, that's kind of cute. Kind of can
and
move
you
on.
can also look up GIFs too. So like on YouTube, like you'll see like videos that people have made of proteins and like how they look when they're like functioning. That was always like
great.
My favorite. Yeah, those are so
great.
fun.
Oh, yeah.
Yes.
Oh, yeah.
Proteins are cool.
Proteins are cool. They're fun to look at. DNA, RNA, they're cool looking. They tend to have a few structures that you can look at, but proteins will look like anything. You can get proteins that really have any shape, size, maybe not size. There's no proteins that are big enough to see, but there's, well, I don't know strictly how you define that. Your hair is made out of proteins, all kind of linked together. It makes your hair do hair things.
I hope there's a protein that looks like me somewhere in the world.
I'm gonna go to AlphaFold and be like, can you, hey, make a protein that looks like Maia.
Ooh, that would be
fun challenge.
That would be a fun waste of massive waste of computing resources.
A fun waste of AI.
I mean,
what
hey,
else
I mean, if I could ask it to make a picture of a duck in a party hat, I could probably ask it to figure out the protein sequence that folds into our friends.
Yeah,
that would be fun. I feel like people have done something like this. I feel like it's out there. We should search for it.
The reverse of put in the sequence to get a structure is hard. It's not like a thing I've heard of anyone doing successfully with like function, at least, without starting somewhere. But if you're not looking for function, if you're just looking for structure, if you're just looking for blob, 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 maybe you can get it.
I do think that a Maia-shaped protein would probably not be the most useful thing as ATP synthase rotors or like...
I disagree. You don't know that.
I'm just floating in.
Yeah. Maybe there's a Maia-shaped protein that cures cancer.
Whoa. Whoa.
I doubt it.
I knew I was onto something.
When you were born.
Just Maia shape, not Natalie shape, not Sam.
No,
just specifically me.
Oh boy! All right, it's only Monday, folks. We're recording this right before at least right before the week before like the holidays that I will be celebrating. But
Same.
It's probably gonna come out after that because frankly we're not gonna probably force Pickell to edit it and everything. So sorry in advance for the belated episode, but also I'm sure y'all are busy too. So
and also just want to say little sappy here, but this is our second full year of doing monthly episodes, really sticking with it. So I just want to say shout out to the listeners who have been here, the new listeners, the old listeners. Shout out to the Interactome team. I love you guys.
No, thank you so much. Literally everyone. Yeah. Thank you so much.
Thank you. It's amazing. People actually listen to this.
I know. Don't know why.
No, my roommate was like, I listened to your podcast, and I was like, oh.
Here, don't, please. I'm embarrassed.
I've got a part where it's like, so I have this science podcast. I'm like, wait, people ask. I'm like, okay. It's I'll be terrified the day that somebody hears about us from the podcast. Hasn't happened yet.
Like in the grocery store, I know your voice.
Oh no.
I'll run. I'm out of there.
Yeah, I don't know. It's not gonna happen for a while, probably.
Yeah.
But listeners, if you want to make/ruin our days, share this episode. Tell your friends. Keep doing that, and maybe someday Natalie will run out of a supermarket screaming.
All right, bye everyone.
Bye.
Bye, everyone.
You know where to find us.
Hopefully. And if not, we are some variation of the Interactome or Interactome Media on your social media of choice. And even though we're not always super active on there, if you message us, we will be delighted and we'll probably reply. So anyway, hope everyone has a good winter break, holidays, whatever. I guess we'll have had a good break, holidays, whenever this comes out. Anyway, have fun, or I hope you had fun. And thanks for listening.